Hi everyone. Welcome on a lovely autumn afternoon to another Genomics Aotearoa seminar. Today's seminar will be provided by Paul Gardner from the University of Otago. Paul's been around the bioinformatics scene in New Zealand for a long time, and many of his publications are extremely well worth reading, particularly the one about trying to identify how enhancer elements evolve in birds, which I think is a bit of a killer. So I think without further ado, I'll let Paul present on his topic, should I be trusting my bioinformatic tools? Go for it.
Kia ora koutou, ko whoretu tuaka no whangara ahu. Kia ora people, and thanks for that kind introduction, Peter. So yes, my name's Paul Gardner, and I hail from a very small place just north of Gisborne called Whangara. But more recently, we've been quite happily living down here in Otago. And William sort of scuppered my first joke for the talk in the chat message, and that my talk title is "Should I be trusting my bioinformatics tools?" And yeah, I could probably just answer no, you should not, and just stop the talk there. But nevertheless, I'll try to truck on and try to justify why we think we should be a little cautious about what methods we're using, even if the methods are computational. Hello, it's not a good start. Let's see, there we go. Okay, so scattered throughout my talk, I'm going to have lots of photos of the beautiful Otago coastline. So during my weekends, I generally take the opportunity to go out on the water and explore this. So if you're getting really bored with what I'm saying, hopefully you'll at least have something to look at. So this was a fantastic day last year where I took some of my kids exploring some of the longest sea caves in the world that we happen to have just north of Dunedin here, within the Matanaka cliffs here. So I think we've got something like 6 out of the top 10 longest sea caves, including the longest sea cave in the world, which is pretty awesome. And on this particular day, when we finished caving, we came out and we were greeted by a huge pod of dusky dolphins. So that was a pretty cool day. Anyway, so when we're not kayaking, my lab is generally pretty interested in things related to RNA biology. So we spend quite a bit of time trying to work out how can we discriminate between transcriptional noise and bona fide functional non-coding RNAs there. And so we've got a number of strands of projects that are looking at that. We look at things like evolutionary conserved structure, interactions, accessibility of RNAs as well. We've been collaborating with a group up in Auckland led by Austin Ganley on a random genome project, which is pretty cool and lots of fun. And another project that we've got running is looking at the mutational robustness of RNA and protein. Some of the other things that we look at are analyzing genomic variants and working out which ones are likely to be functionally important and which ones are probably neutral when it comes to phenotype. And then finally, we've got this broad interest in which bioinformatic tools are actually useful for some of the tasks that we might want to solve. So we're often faced with a challenge if you want to do something, so analyze some data within, say, genomics or the realms of bioinformatics. So we often have lots and lots of different software options out there. So one of the first things you often want to do is work out what is a good tool. So I thought I might ask for a little bit of audience participation here and just see if anyone's willing to share, how do they choose software when they're faced with lots and lots of different options there. And I might pick on Martin Kennedy at this stage. I think I saw his name somewhere.
So kind of you, Paul. I not being a bioinformatician, I usually ask one of my clever students what software I should be using, or they should be using for the dumb question I just asked. But I'm also aware that you exist, and I'd probably ask you as well. So
Right. And I see someone else has done almost the reverse. So they use what their supervisor tells them to use from the chat. So thanks for that, Zhuhong. Okay, so you just ask a student. Do you have any ideas how your students select software?
me in particular, Paul.
Yes, I'll keep picking on you, Martin.
Yeah, they're probably influenced, particularly. I don't know if John Pearson's online, but hopefully they were going to have a chat with John Pearson. But we tend to use stuff that's pretty well established and not really developmental. Although Oscar in my lab is a pretty good bioinformatician, so he will pull things together and do a bit of coding and stuff. But yeah, it's a bit ad hoc. I don't think we have any clear rules.
All right. Oh, very good.
That's probably comes from me.
Okay. So we're getting some quite useful comments in there. So I think someone may have been flicking through my slides, or so Adam suggested good documentation and recent GitHub activity, which is pretty good. Volta suggested publication impact. And yeah, citations are fairly commonly used there. So I might truck on. Ask colleagues who know better. Cool. Or, find a similar paper and just copy what they do. So those are fairly common strategies for selecting software. So do you know or are you some way connected to the developer? They might be a famous, really high profile scientist within the area. You could identify the most recently published one, which I think someone has suggested. Is the software tool widely used? So, if you've had a bit of a browse through the literature, are one or two tools being used fairly often? People often look at the journal that things are published in as well. Is that a high impact sort of Nature or similar journal there? And I've heard of a few people looking at software speed as well, and often doing the reverse to what you might expect. So some individuals think a really slow tool is likely to be very accurate, which I found to be quite an interesting idea. All right. So one thing I'm going to advocate for is that we try a whole bunch of different tools, but develop like a good set of controls to run those tools on. So find some positive examples of whatever you're trying to predict, find some good negative controls for whatever you're trying to look at as well. Run the tool over those and see if they discriminate cleanly between those data sets. And that's kind of what my group has been doing for quite a while now. So we started out looking at RNA bioinformatic tools, looking at secondary structure prediction, homology search, and sequence alignment for those. And the fun thing about doing that is you can identify what are the really important factors or good ways to make predictions for each of those problems. We've also looked at eDNA tools as well. So basically sequencing lots of environmental DNA and try to work out, where on the taxonomy that DNA may have come from there. Recently, we looked at bias in self-reported accuracies of tools. So it turns out the developers usually like to publish their tools in the most glowing light. And we're going to sort of circle back to that idea as well. So often the self-reported accuracy of that developers have reported is very different to the accuracies that independent groups will identify for different tools as well. And recently and if there's time at the end, I will we'll discuss some of the meta-analyses of benchmarks that we've been looking at. But today we're going to look at coding tools. We're going to focus on tools that will take, say, transcriptome data or genomic data and work out whether the sequences contain coding sequences or non-coding, not protein coding. And then we sort of leave it as an exercise to work out whether non-coding sequences could be a functional RNA or a, noisy transcript or just some random part of the genome. There. Okay, and so what led us to this project was a fun collaboration with a group at Plant and Food Research there. So a team led by Samantha Baldwin, together with Rebecca Bloomer, Susan Thomson, and Ting-Hsuan, based at Plant and Food, sort of recruited me to help out with some of the analyses there. And I recruited DJ Champion, who's based down here at Otago as well, for this project. And the project was to sequence transcriptomes for lots and lots of different Solanaceae species. Lots and lots, several Solanaceae species. So tomatoes, potatoes, what else do we look at? Eggplants, black nightshade, and we were hoping to look at poro as well, but didn't quite get to it there. The idea was to take different tissue types from those, look at the transcriptomes in each of those tissues, and identify the long non-coding RNAs within those data sets there. And one thing that Ting-Hsuan discovered when she was analyzing that data was there was a big difference between the homology-based coding annotation methods and then the purely de novo sequence-based annotations as to whether these transcripts were coding or not. So hopefully that makes sense. So the homology-based methods basically take a protein sequence database and align those to your transcriptome data there. And if it happens to match a known protein, you then sort of class that sequence as protein coding. If it doesn't match, then you classify it as a non-coding sequence. But of course, we've got sort of de novo purely sequence-based methods for trying to determine that as well. And I had thought this was a pretty mature field. This is a very old problem. We've been sort of, trying to predict this, since the very early days of sequencing as well. And so I had assumed the tools were pretty mature for looking at this problem. But when Ting-Hsuan mentioned that there were some pretty big discrepancies between de novo methods and homology-based methods, I thought we might dig into this a little bit more. And so I asked DJ to start putting together some test data sets, so some positive control data sets and some negative control data sets to look at this in a bit more detail. We also recruited Mick Black to get involved in this project as well. So Mick has been really useful at helping out with some of the analyses on this. But I also thought Mick would be a bit of a moderating force because I thought I was getting too angry when I was writing up the paper and just being too mean to the authors. But if anything, Mick has made it much, much worse because not only can we say some tools are quite bad, but we can also talk about how significantly bad some of these tools are likely to be, thanks to his input. So basically, so the first thing to do was to identify what are some tools that we can assess for this method. So we ran some iterative literature searches, looking at reviews, and mined the references and citations for those. We looked at some existing benchmark data sets as well. And we identified around 36 different tools that we thought might be applicable. And then we assessed them against these 7 criteria there. So is the primary purposes of the tool to predict whether something is protein coding or not from a nucleotide sequence or alignment, was sort of the main criteria that we were looking for. And then we had these additional criteria. So is the tool available? Was one. Could DJ install the tool? Now, DJ worked as a system administrator for quite a few years, so he's actually really good at installing stuff. So that shouldn't have been too much of a problem for him. Was the tool generalized, or did it only run on say, a certain group or a certain clade of species? So, if it only ran on, say, plants or only on fungi, then we excluded that method as well. Then we had a few other criteria as well there. What was also kind of disturbing for me is that lots of tools were not accessible. So around 20 of these 36 tools, around 20% of these 36 tools were just not available on the internet. So we couldn't download these software packages. Then almost a third of the tools could not be installed once we, had downloaded them as well. And then about a quarter of them were specific to one certain group. So we excluded them. So after we assessed each of these 36 methods, we were left with 9 tools that sort of met all of our criteria and that we could include in our assessment of software. And some of these tools are really widely used as well. So CPC2 is cited about 200 times per year. I forget the exact numbers, but the 2 papers describing this method have thousands of citations to them. And then, sorry, I might move the mouse, so I stop touching. Oh no, I still need the mouse. Okay, something strange happening with my keyboard. Right, so the next most highly cited tool sort of accrues nearly 75 citations per year as well. This is a comparative method, so it looks at alignments there and looks at the ratio between synonymous and non-synonymous changes to predict whether something is coding. And then, I'll sort of look down the bottom here. So this is a very new tool. So it was only published a few months ago in PLOS Computational Biology. So it hasn't had any citations yet. And then these 2 methods were methods that we created for this benchmark as well. So now those 2 methods were meant to serve as baseline methods there. So we wanted to develop some very naive tools that should serve as sort of the lower bound on how well a coding prediction tool should perform. So the first method that we call stop-free just looks for the longest run of stop-free sequence in any of the 6 reading frames for any input sequence. Okay, so hopefully that makes sense. So you're just looking for a run of sequence that has no stop coders. So no UGA, no UAG, or no UAAs in any of the 6 possible reading frames for a coding sequence there. And we thought surely all the tools should do much better than this method because it's very basic. There's no machine learning there. Then the other baseline tool that we used is we call it RANScore, and it's a random number generator. So it generates a random number, basically just selects one of the integers between 1 and 100 there, and that's the score. Then, so we've put together the summary table of the other methods that we've included as well, of the 9 tools that we include. So 7 of those were purely sequence-based methods. So you input a sequence and it tells me whether it's likely to be coding or not there. And then 2 of the tools use alignments. So given a sequence alignment, are these sequences evolving more like a coding constrained sequences, or are they fairly unconstrained or potentially a non-coding RNA? Is what these 2 tools do here. Now we got a little bit cross when we discovered this, is not all the tools run across all 6 frames there, and many of them don't even report what frames they look at. So some of the tools explicitly tell you they're scanning across all 6 possible reading frames for any input sequence there. But many of them don't document that. And so what DJ had to do was generate sequences in all basically 6 reading frames for each of these tools. So basically trimming off either 0, 1, or 2 nucleotides from the beginning of a sequence, reverse complementing it, and then doing the same thing on that reverse complement. And then running each of these tools across all these methods, and then we could evaluate by looking at the scores from these tools to work out how many frames they were actually screening or not screening. And it turns out lots of them are only looking at the forward strand there. There was one tool that we ended up removing that only looked at 1 frame as well. So it just assumed that we had given it the correct frame there. So I've chosen to name and shame those methods in this table there. DJ has also assessed how easy the methods were to install, how easy they were to use, and how useful the output for these methods is there. And you can see, for many of the tools, installation was not too bad, but for some of the tools, installation was horrendous. So I think PhyloCSF, for example, depends on some decades old piece of software that you have to go and find and install, which takes quite a bit of work. Many of the tools had no installation instructions. Many of the tools had no documentation about how to use them. They had things like undocumented parameter options. The outputs were not terribly well documented, and often the error messages could be quite uninformative as well. Hopefully I've captured most of the issues that DJ stumbled across. But he basically, against each of those 3 criteria, gave things either a smiley face, a fairly grim, acceptable face, or a sad, this took a lot of work to get going face there. And, oh my goodness. This mouse is very sensitive, sorry. There. We've also recorded the versions of each software tool and provided references for each one as well. Right, so then the next thing we had to do was to assemble some test data as well. And what we've chosen to do is identify well-annotated and well-assembled reference genomes that had never been used in the training of any other coding or any other coding prediction tools that we were looking at. And so what DJ has done with those is sampled exonic sequences from those, and then matched those with neighboring intergenic sequence as well that had not been annotated there. So we're presuming those intergenic sequences are not protein coding, but the odd one may have been missed by annotation tools. But we're assuming that's relatively rare for this project. He's also included some flanking sequence for each of those, just so we don't make it too easy for any of the tools there. And yeah, we ended up selecting the cat genome, Felis catus. We ended up selecting the melon genome, so Cucumis mello as well, and an Aspergillus genome as well. So we've got mammals, plants, and fungi represented in this data set. And we sample about 1,000 exons from each of those. There, we've also carefully batched those with additional genomes that are fairly closely related, but at different taxonomic distances there. And we're going to use those to build sequence alignments for the comparative methods there. So for, oh my gosh, this is not going well. There we go. So for the positive sets here, I've just annotated an example here. There we, I've kind of marked up each of the codons within this alignment there. What is the amino acid that each codon encodes? And then you can see, some of these codons contain synonymous variation. Hopefully you can see there. So in this first triplet here, each of these sequences encodes a valine amino acid. The next one along, each of these sequences encodes a proline, and so on and so on, across these 4 different species there. For each of those, we also shuffle the alignment as well, and that serves as a further negative control. And then each of the intergenic sequences, we also make an alignment for those there. Okay, and then we can feed all of those either alignments or sequences into each of these tools and see whether they can correctly separate the positive sets from the negative sets. This is the basic idea. So this is some of the results. So I'm guessing not all of you will be familiar with ROC curves. So a ROC curve is basically plotting the specificity along the x-axis, or in this case, 1 minus the specificity. This is also called the true negative rate there. So how many of the things that you say are not coding are actually not coding. And then you plot that against the sensitivity, or the true positive rate. So how many of the things that you say are coding sequences were genuinely coding there, is what you're plotting on the y-axis there. And then for every possible threshold, score threshold for each of the tools, you can plot a point. And each tool has a trajectory through this plane here. And the perfect tools have a perfect specificity and a perfect sensitivity. So we'll pass through this point, at the top left. So we'll have 0 as the 1 minus specificity and 1 as a sensitivity there. So none of the tools were perfect, but RNA code did particularly well in this test. Next down the list was phylo-CSF as well. It has this weird dog leg, which we don't quite know what to make of yet. We may have to dig into that a little bit further there. But those 2 methods are both of the comparative methods. So it may not be too surprising to people that, if you look at the evolution of a sequence, you can be much more confident in your prediction. Now down the bottom here, we've got our random number generator, and that basically marks a diagonal across here. So that's pretty much what you would expect a random number generator to do on this sort of plot there. And weirdly, this tool, LGC, follows the random number generator fairly closely there. And I'm going to talk a bit more about that later, but it seems almost as good, only slightly better than a random number generator on this particular data set, which is a bit worrying. This other tool, CP-Pred, is fairly close to that as well. Well, slightly better. All right. So just looking at the accuracy data in a slightly different format. So this figure's a little busy, which I apologize for, but what we're showing here are 4 different accuracy metrics for each of the tools there. And each of the points here corresponds to one of those 3 control data sets. So either the mammalian one, the plant one, or the fungal group there. And we've just recorded the mean area under the ROC curve is a fairly common accuracy measure. The sensitivity and the specificity for each of the tools, and then a Matthews correlation coefficient as well. So if you're perfectly correlated with the truth, you have an MCC of 1. If you're a random number generator, then your MCC should be around 0. And if you're perfectly reverse correlated with the truth, then you have an MCC value of -1 there. Okay, and the other thing that we're showing on this plot is the self-reported accuracies for each of these tools. So these black crosses and the arrows indicate the self-reported accuracy that tool developers have shown in their papers there. And you can see, who should I pick on? So phylo-CSF, for example, reports a specificity of almost 100%. in their paper. And I think their, what's that measure? The sensitivity was also in the high 90s that they reported. Whereas we're finding values much, much lower than that for many of these tools there. So BioSeq2Seq, this recently published tool, for example, you know, they're reporting MCC values and sensitivities, again, in the high 90s. So very high sensitivity in MCC, but in our hands, performs much worse. So the MCC was down about 0.25, and the sensitivity was, what's that number likely to be? Around 60% there. So we're seeing some fairly huge discrepancies there. In fact, the tool with the worst self-reported accuracy was RNA code. And it turns out that their accuracy values that they reported were pretty close to what we were finding for that method as well. So, you know, if you're going to be reading, you know, these tool papers or these descriptions, I'm not sure how much we can trust the self-reported accuracies for each of these methods, which is a little bit unfortunate. So this was a suggestion that Mick had as part of this analysis. So with the ROC curves, we can work out how significantly different different tools are from each other. So what we did here was we took our stop-free method. So this was really naive, no machine learning involved, just a single length metric there, and worked out how significantly different each of these methods were, either in the positive direction or in the negative direction for these tools. And what is kind of surprising for me is that only 3 of the tools were significantly better than the stop-free method that we reported here. So the 2 comparative methods and this T-code method as well. And what T-code is an implementation of the Fickett score, which was published more than 40 years ago. And way back then, basically what they did was they developed a probabilistic method based on nucleotide frequencies that they extracted from what they had then were 321 protein coding sequences and 250 odd non-coding sequences from fairly diverse sources. So viral, bacterial, human, and yeast sequences there. And that turns out today to still be one of the better tools on this data set, which is a little troubling. All right. So, all right. So this is probably one of the pettiest figures I've ever made here, but what I've done is I've classified each of the tools in terms of what is the machine learning method that was used, and then also what field the group that published it comes from. And that was just based on the departmental names there. I might be a little sensitive about some things. I had some harsh reviews on my recent grant application there. So I just want to point out that, you know, you might be an expert in, say, machine learning or computer science, but it doesn't necessarily mean you're going to write fantastic software for these sorts of problems. There. All right, so some of these tools are using fairly sophisticated machine learning methods. So things like neural networks, support vector machines were fairly popular a while ago. And maximum likelihood approaches seem to be covering sort of both ends of the spectrum. Whereas RNA code, which is, you know, our best performing tool there, was developed within a bioinformatics group there and is using quite a bespoke dynamic programming method to look at insertions and deletions. It's looking at synonymous versus non-synonymous sites. It's also looking at how within the non-synonymous changes, how conservative are those changes as well. So using a Blossom score matrix to identify those. And it's also fairly careful to try and model potential misalignment and things like that as well. So there's quite a lot going on with that method. All right. Okay, I think that's all I better say about that. So in terms of speed, we had thought that the alignment-based methods would be the slowest, and it turns out phylo-CSF is indeed one of the slowest tools there. So it can only cover, what is it, a few hundred nucleotides every second if you were going to use this for any sort of scanning, which is not great. But then the next slowest tools were these neural network based methods, so bioseq-to-seq and RNA-Samba, which were not great. And then you might remember CPC2, which is one of the most popular methods, is also fairly slow in our hands there. So only, screening, say, 1,000 kb or so per second. RNA code, which is doing a lot more, is roughly as fast as that there. And then these very simple simple methods are essentially linear in terms of their speed. So T code is about the fastest. Remember, this was a tool written 40 years ago as well, so we think that's pretty good. Now, hopefully you can remember LGC was performing really badly in this particular data set there, and it turns out we hit a fairly major weak point in LGC. So LGC is based on the length of an open reading frame. They don't really say this in the paper, but presumably they're looking for a canonical start codon, so an AUG, followed by a canonical stop codon as well. And so it's sort of modeling the length of the longest open reading frame in the input sequence and comparing that to the GC content as well. So it's only using two parameters there, and then using a maximum likelihood prediction based on those two features there. Now, the melon genome, it turns out the intergenic sequences are really low GC content, so around 28% on average there, whereas the coding sequences around closer to 40% there. And if you look at these figures for the solanum and rice examples that they had in the LGC paper, you see there's a fairly significant overlap in the coding and non-coding sequences for both of those genomes in terms of the GC content and the auth length there. And so it looks like there's a big sort of weakness in this method for low GC there. And it looks like the scoring scheme essentially flips there. So it was assigning very high scores. Actually, I can show that on the next figure there. So the intergenic regions all get very high scores within the plant data there. So we've normalized all the scores here, and we're just using box and whisker plots to show the results within animals, fungi, and the plants here. You can see within animals and fungi, there's a bit of a shift in the score to higher values within animals and the fungi. And ideally, the method would have scored all the coding sequences much, much higher than our non-coding control sequences there. Within the plants, it's scoring those intergenic sequences, the low GC intergenic sequences, really, really highly. And so basically that's producing results that are fairly similar to our random number generator control just because of that. They might have been better off just not reporting any scores at all for low GC sequence there. So how has this field gone so badly wrong? And I think one of the issues that we identified when we were summarizing all of these different tools is that most of them rely on RefSeq as a source of positive coding sequences and GenCode as a source of non-coding sequences, but mostly using either long non-coding RNAs or short non-coding RNAs as their negative set there. And so what we need to keep in mind is both of those data sets are highly curated. You know, they've had decades worth of curation for some of those data sets. And so they're fairly free of any sequencing error, any assembly error. There's almost no truncated sequence going into those data sets either. So they're getting a really false view of how clean either transcriptome data and genomic data is likely to be. And so if you're running generating your own transcriptome data sets, it's going to be a lot noisier than anything in either of those databases there. And what these tools have done is sort of trained on really clean data there, and which means that they can't perform well on noisy data, more realistic data. The other issue that we have identified is there are some huge differences in the lengths between the messenger RNAs in those databases and the non-coding RNAs in those databases. So what these tools could have done if they wanted to perform, okay, is just use the length alone as a predictor there for those tools. I think they over-rely on canonical open reading frames as well. So most of them require a canonical start codon, so an AUG, and canonical stop codons. But with truncated, sort of messier data sets, that's not always going to be the case. So I think these tools need to be much more robust to truncations, frame shifts, any sort of sequencing error there needs to be catered for. The other thing we discovered is that homology between the training and test sets was generally not controlled for as well, which means they're sort of biasing their assessment of the accuracies as well in a lot of these cases. If you've got very similar sequences in your validation or test set compared to your training set, then you're going to inflate your accuracy scores there. And then, I also wanted to mention that many of these tools failed to document how many frames they're reporting, whether they were sort of a forward strand only type of method, looking at all 6 frames or just a single frame there. And a worrying number of the tools did not report the coordinates when they found a coding sequence, which makes it really difficult to validate some of these tools. All right, so some other conclusions. I don't think we can trust self-reported accuracies in general. They certainly were vastly different between the self-reported ones and the ones that we measured. These tools are probably fine on perfect data sets, but if you've generated a perfect data set, you probably don't need these tools there. And there's probably plenty of other things that we could do in this space as well. So we could look at more diverse species, so source data from, say, organelles, protists, viruses, bacteria, etc. There we could generate our own transcriptome data sets as well and run over those. And we could look at, simulating data sets with, say, sequencing and assembly errors, generate truncated data, things like that as well for more realistic assessments as well, in addition to what we've already done with the exonic data. So I think I've just about run out of time. I was going to mention some other work that we've done, running a meta-analysis, looking at some of these questions as well. But I'll skip over that just to say that things like impact factor, how famous authors are, turn out to be really bad ways to predict software based on our analysis there. Turns out the better way to identify things, and I think someone mentioned this in the chat, is to look at activity on GitHub, look at the versioning or version control and version histories for software tools. It's not a perfect predictor, but it will give you a much better idea of whether a tool is likely to be good or not. All right, so I think I'd better finish there. Hopefully Peter hasn't gone to sleep in the meantime.
No, I woke up in the middle. That was really fascinating, and people, please feel free to unmute and ask a question directly, or put a question in the chat.